when you'll also have to study for exams and stuff like that. So this week we will start biotech, and biotech is actually not covered on any of the actual exams. It's covered instead on this biotech lab exam. So I'll talk a little bit about that, and let's just look at our kind of the schedule for week 5. Okay. So for this week, as I said, we really only will have the biotechnology material, and then also we'll start cell cycle. So those are the 2 ICAs that you'll have for this week. The lab starts off with basically setting up the RNAi. And the reason I had a delay in getting this up online is I kind of had to think about how do we want to do the RNAi treatment. So as we know, as soon as we put the worms on the food, they're going to start eating the bacteria that has the plasmid, right? And if we add IPTG, does anybody remember what will happen if our RNAi plates have IPTG in it?
Yeah. So IPTG will stimulate the operon to create the T7 limerase,
Yes.
which will bind to the L440 T7 promoter sites, creating a double-stranded RNA.
Yeah, that's a great explanation. So essentially, as soon as we add IPTG, the double-stranded RNA will be created, which will start inducing RNAi. So the question that I had in my own mind was, when should we start knocking down the gene expression? Remember that with RNAi, you don't affect any proteins that is already there will not be destroyed. So it'll still be in the worm. So one possibility is to basically treat our L4 stage. So if we start IPTG at the stage when they're at L4, we could then take those L4s and put them to the high temperature. The problem with this is that they haven't had a lot of time to stop producing the gene product. And whatever protein has already been made will still be there. So this method right here, treating at the same time point as you're shifting them to higher temperature, may not work. So we're actually going to do it one generation where we are going to feed L4. Now L4, do they have any eggs inside when you look at them? No, they haven't yet developed. They actually haven't fully developed the gonad. So at L4 they are immature, but if they start eating this when they're producing their oocytes, when they're producing their embryos, then the embryos will have no product, whatever gene you're knocking down. So let's say we start them at L4, they grow to be adults. So I'm going to show that they're adults, they have eggs. These progeny, this is the next generation. You would say that this is the F1 generation. This is the P0 parental generation. F1 is the next round. Those embryos are the ones that have never had that product. It's never been present for them at all. So that's why we are drawing out this experiment over 2 weeks. And we're going to basically feed L4s, but we're going to test not that L4, but we're going to test the next generation, which means we have to let the L4 have the embryos, and then when these embryos are laid out onto the plate, they're going to hatch into L1, and then they're going to go through all the stages, L3, L4. This is the one that we're going to then shift. So it's kind of hard we have to do the best we can to estimate when that's going to happen. When will we have L4s from that generation ready to go? But I did the best based on kind of what generally is the kind of time scale. So our first step is to make sure that we have L4s ready to go onto the plates. So remember when I gave you plates and they had a little chunk on them? So chunking is one of the ways that you can propagate lines. And so essentially if you have a plate that is starved out, on that starved out plate there will be dowers, which does anybody remember what the dower is? Yeah, Daphne, you remember?
Dower is right when they don't have enough food or something, they'll kind of go into the
on the food they re-enter. So we will take plates right today that have dowers that are starved out, and I'll show you how to chunk from that plate and start a new plate. And that's the plate that by Thursday, hopefully, that plate will have L4 that we can then put on to your RNAi plates. So there's kind of two things we're kind of doing in the early part of this week. One is that we have to restreak your sequence confirmed RNAi clone. So remember how we made glycerol stocks? That glycerol stock came from a single colony. So we're going to go back to that stock, and you're going to restreak it out. Does anybody know why we would want to streak from that glycerol stock instead of going back into the library and restreaking from that well? Anybody know? Yeah, go ahead.
The one in the glycerol stock we know for sure is a monoclonal colony, versus the ones from the Eringer library, there can be different.
Yeah, exactly. Yeah, so the one that you selected, you grew it up, and then you sequenced it, you confirmed that it was what you thought it was. So that is trustworthy for us. So that's why we're going to go back to that stock. So does everybody remember how to do the streak to a single colony? We'll be doing the same thing. You won't have to use a needle today because we're not going into a tiny well. You can just start with the pipette tip. But again, you'll do the same technique. You're going to streak and then change the tip, streak again, change the tip. And the goal is to get it down to a single colony. So those two things we'll do today. So as you can tell, that's not going to take a ton of time. So I will give you time today to work on the introduction for the posters. And then tomorrow, hopefully we get the plates out of that incubator. That's our 37-degree incubator. And hopefully there's just a single colony so that you can pick one of those to start an overnight culture for actually putting onto the RNAi plates, which have IPTG, and they also have ampicillin. The ampicillin helps to select for the L440 plasmid, make sure that only those bacteria are able to grow. And so you'll be basically seeding the plate so that you give the worms food to eat. And when you do that, that's going to start producing double-stranded RNA. The next day, which will be Thursday, is when you're going to actually put the L4s from the chunked plates today. They hopefully will have some L4s. You're going to be putting those on to the RNAi plates. Okay? So one other thing, because I got some emails, and I know there's a lot of confusion. We never discussed it in class, and I don't expect you to just know this randomly, but when we set this experiment, each group has their own clone that they're going to be testing. And if we think about it, you're going to test that RNAi sample. We have to test it on N2 and GMC 101. Does anybody know why we still need to test the RNAi on N2, which is our control? Any ideas about that? Yeah, do you want?
The N2 does not have a paralysis phenotype, so we have to see if there's actually any difference with having the plasmid knockdown with a paralysis phenotype or without.
Yeah, exactly. So this one does have a paralysis phenotype, this one does not. And so we're going to see if when we add in the RNAi, does it have its own, maybe it has some kind of phenotype on its own without mixing with this GMC 101 background. So testing it on N2 will let us see if that is the case. For simplicity, I think what we're going to do, if you went back to the data, for 16 degrees, did anybody observe significant paralysis on either strain? We didn't find it. So if you wanted to set up the most robust assays, we would test, 16, 25, N2, GMC 101 with the control, and also with the RNAi. So we would test all of these on each sample, but I think for simplicity, because we did not observe it at 16, we're just going to test the shift in temperature, just for simplicity's sake. You're still going to have N2. You're going to have N2 with the negative control, RNAi. And I wrote in the notes for the lab, I said that we're going to use SMD1. So let's just talk a little bit about what a negative control is versus a positive control. Does anybody kind of have a sense for the difference? Negative control versus a positive control? Yeah.
If I remember correctly, positive control is something that we know is going to happen, whereas the negative control are kind of like,
Yes.
maybe this is.
Yeah, exactly. So negative, so in different experiments, they're gonna be different things, but the negative control should not have, it does not contain the variable, I guess, that you're testing. So, it should not have a phenotype, I guess, in this case, where we're testing, for example, when we just had N2 in GMC 101, the N2 was the negative control, because we don't expect us to have a paralysis phenotype. For the RNAi though, we have to somehow initiate RNAi, but we initiate it with something that we don't expect to have a phenotype. So in my postdoc lab, we always use SMD1. If you look up SMD1 on worm-base, it does have some phenotypes that are post-embryonic phenotypes, but we always use it as a way to control because it doesn't have like lethality as a phenotype, it doesn't have paralysis as a phenotype. And so we know we're initiating RNAi pathway, but we don't expect it to have any effect on, certainly on paralysis, okay? For a positive control, so you said the positive control should have some phenotype. So there's some different things we could do. We could, if we wanted to have a positive control that had a paralysis phenotype specifically, because we are looking at, we would use something that had been confirmed to have a paralysis phenotype. Currently, I haven't verified any of those yet. So we're going to use a positive control that causes a known phenotype, which is very severe, and that is embryonic lethality is the phenotype. And it's very easy to score this because if you are lethal at the embryonic stage, it means that the eggs don't hatch. So it'll be easy to look on a plate and see there's no little tiny worms. So this RNAi must have worked. What that tells you is like all of your reagents are good. The IPTG was working. Everything should, in theory, work. So that when you test your construct, in theory, the conditions have been right for this to work. So then you should say, okay, our RNAi should be able to work. Does that make sense? So in an ideal situation, I would say we would pick something that had a paralysis phenotype. But in absence of us having confirmed that, we're going to do one that has been confirmed by some of our capstone students to have embryonic lethality. And the gene, I think it's called PFN1. I have to make sure I can find that glycerol stock. But this is profilin. So you want to write this down in your notes. This is our positive control. This is our negative control. And this one should cause embryonic lethality. So when we put those L4 worms at this stage, we put them on profilin 1. When they lay the eggs will never hatch. That's how we'll know that that worked. Okay, does that make sense? Okay, so as a class, we are going to do, let me move this down a little bit. We're going to do one negative control, one positive control for both, this and this. And then you personally in your group will be responsible for doing your chosen RNAi, whatever that is. So for you actually will only be setting up the two plates, the RNAi with N2, the RNAi with GMC 101. And I'm gonna set up negative control, positive control for N2, and negative control, positive, just so that you can pick and it doesn't take forever.
Until late at night.
Yeah, does that make sense? And so for these ones, everybody will be able to look at them and score them. And we'll kind of, like we did last time, we'll put our data up on a class data sheet so you can see, everyone can take a look and share the data that they find. All right, is there any confusion? Because I know Lab 8 is extremely involved, but is there any confusion about what we'll be doing? So this week is all set up. Next week is when we actually will do the shift to the higher temperatures. And the other thing I will say is that when we put them on RNAi, we'll put them on RNAi on this day. We'll actually shift them, which I may have to help out with if it happens over the weekend. But it's a good idea to keep the food fresh so that they keep getting like renewed function RNA trigger. So I'm gonna have you set up double the amount of plates that you actually need for RNAi, so that we can kind of do a transfer.
So you're saying we're seeding a
Yeah.
total of 4 plates?
Yeah, well, so let's think too, these plates will be, the L4s will go on there. We might transfer them, and so you might actually seed 3 because you're gonna do the shift. You know, you're gonna shift them to higher temperature. So, and we still want them on the RNAi food plates on that day as well. So
So we're doing the chunking on
regular
a
plates.
regular plate and then taking L4s to the RNAi plates.
Yes.
And then taking their progeny to another RNAi plate.
Yeah.
So, okay.
And I'm gonna throw in 1 extra, so that's why you'll do 3,
Okay.
just so that we kind of refresh the foods. And so they're always eating it, and then hopefully these ones that are born, they've never produced that food ever, or they've never produced that gene product. We'll see how it goes. This is the first time we're doing this experiment, and we'll just see. So then you'll be kind of comparing your GMC 101 to the negative control, but you can also compare your N2 RNAi sample with your GMC 101 RNAi sample. So you'll have a lot of data to kind of deal with next week. Okay, so then the other thing, this Thursday, we're gonna do this authentic research project, which is over all these labs. And so I'm gonna put together a study guide tonight so you because I know that with this especially, there's so much stuff out there, it's hard to know what do I need to really have mastered for this. I do have a study guide for biotechnology that's already on Canvas for that part of it. This exam is not as big as a regular exam, it's just 75 points instead of 100. And also, what I did last year, I think last fall, people liked, is that you do the exam yourself, so you can have 1 hour to complete the exam, and you turn it in, and then as a team, you take the same exam again. And I think I did last year, basically 2/3 of your grade comes from your individual score, and 1/3 comes from the group score, so you can raise your grade. I never lowered it, so like if somebody did worse as a group than they did as an individual, I just gave them the full score that they got. But anyway, it gives you a chance to talk through the ones that you didn't know. And in that case, you all turn in 1, so you come to agreement and turn in 1 group exam. So if you all want to do that, we can do that for that lab exam. Okay, any other questions? And I'll hand back the exam from last week. I'll hand it back tomorrow. I still have to grade 1 more. Okay, how did you all feel about that exam? Was it what you expected, or harder, or easier? We need an anonymous poll for this as well. Any thoughts?
It was slightly more difficult than the first exam, that's what I thought.
Yeah.
But other than that, it was kind of expected.
Okay. Yeah, I know that as we progress, it becomes like a lot more process and a lot more details, like a lot more proteins to remember and stuff. And sometimes it's new stuff that you've never seen, whereas the first one might be a lot of review.
I feel like it was just like a lot of information, so it's like hard to study for.
Yeah.
Yeah.
Yes, for sure. Okay. So I'm gonna open up. Oh yeah, I was looking for, I had a great video, but can't find it now. So we're gonna do biotechnology, which in our textbook is chapters 20 and 21. And I think this stuff is kind of it's the stuff we're already doing in lab, so it kind of doesn't make sense to wait so long to talk about it, but it might click with you now that, especially that we have been doing lab stuff. For the kind of the material that I think you should really master is really the first 4 bullet, or the first 4 titles here. So the molecular cloning basics. This is stuff we've already talked about. We've talked about plasmids, we've talked about a little bit about restriction enzymes, you know, as we've been talking about the L440 plasmid. So we'll talk in more detail about how you would construct something, like if you wanted to make your own RNAi vector, how would you do that? We'll talk about sources of the DNA. So if you are going to make your own vector, let's say you wanted to clone the insulin gene into a vector that would go into bacteria to produce human insulin, where would you go to get the insulin source? So the genomic and cDNA libraries, those are 2 places where you can go to get a piece of DNA, and we'll talk about how those are made. PCR, how many have learned about PCR before in other classes? Hopefully, Bio 160 should talk about that. We'll talk about what do you need to set up a PCR reaction, and then DNA sequencing. And we of course already sent our samples off, and they were sequenced. They were sequenced by this method, the Sanger sequencing method. So we'll talk about what they did when they sequenced our samples. And then next-gen sequencing is kind of the new wave of sequencing, and it's why we are able to get whole genome sequencing done so fast. You can actually get your genome sequenced in less than a week. They can sequence all parts of your genome. The last part is kind of just interesting topics in genetic engineering, and that's kind of more for interest rather than like for what I'm really going to expect you to know. So we'll talk about gene therapy, genetic engineering as it applies to like GMOs or foods that we eat, and then we'll talk about genome editing and CRISPR, which you might have heard of those. Okay, so DNA biotechnology really is all around us. I would say that there's applications for this in so many different areas. So forensics is one major area. I have the corn there to say that basically agricultural products, the vast majority of all of our foods, corn and soy, are genetically modified. So we want to understand how do they do that and what types of genes are inserted in to those agricultural products. And then these are actually little, I think they're actually kittens, that have been modified to express green fluorescent protein. So you might think, well, what in the world is the purpose of doing that other than making glow cats? But it's really the idea that if we can figure out how to genetically modify organisms, that we could use that for treating human diseases. Not that we're going to modify humans, but we can create model organisms that we can use to study human diseases. And a very good example of that would be GMC 101, right? It actually is also fluorescent. It does also have the green fluorescent protein. So one of the things we'll look at next week, I'll bring the fluorescent scope so you can see that A beta in the intestinal walls being expressed, or I think in the muscle cells it gets expressed. So anyway, learning about how we do this actually helps us to create models that will then allow us to study many different diseases. Okay, so here are some terms that we need to know. There's the glow cats when they grow up. I think in the interest of time, I put the definitions on this next slide. So for molecular cloning, this is essentially, when we think of cloning, sometimes people think about like Dolly the sheep. They think about cloning a whole organism. But with molecular cloning, we're really talking about how do you move DNA around. That's the core of what molecular cloning is. So it's a set of experimental methods that allow us to assemble recombinant DNA molecules. And I have the definition for recombinant right here. Recombinant DNA means that you're getting DNA from 2 different species and you're putting it together. So for example, in this picture here, when you create a fluorescent cat like this, you're getting the green fluorescent protein. Anybody know where that comes from? GFP? Yeah, it is from jellyfish. So they took that gene out of jellyfish and they can put it into any organism and make it glow. And actually, there's lots of different varieties. You can have RFP, red. You actually can create all the colors of the rainbow and insert it into different organisms.
I want a rainbow cat.
Is that the name?
Well, it actually only fluoresces under UV light, so you have to like hold the UV light up, then you can see it glow. Oh, I don't know. Yeah, I don't know how many are out in the wild, but. Okay, so that's recombinant DNA. The genetic engineering is basically to change, the process of changing the genetic makeup of an organism through DNA technology. I always like to point out, though, in my, especially like my genetics class, that we have been modifying the genetic makeup of organisms for thousands of years, just by selective breeding. So you can do this in an indirect way by choosing two species to mate that, you know, if both of those individuals have traits that you want to emphasize, you can make them together. So we've been doing it, but genetic engineering in this way is just directly doing it. So taking a piece of DNA, putting it in an organism in one step. So that's what we really refer to as genetic engineering. And then transgenic, you've probably heard that used a lot. Trans, I always think I remember, trans is like a cross. So it's taking a gene from somewhere else and putting it into an organism. So it's kind of a way to say that you have added a gene from somewhere else to an organism. This would be an example of a transgenic organism. Our GMC 101 is another example of a transgenic organism. It has the A beta from humans in the work. So that's, and it also has the GFP gene. So it's got two things from two different sources. Okay. So for this, as we go along, we're going to talk, instead of just talking about all these techniques, I like to put it into some kind of a context so that you might think of why would you want to do some of these different techniques. So the story that I'm going to use for this is the scenario of treating pituitary dwarfism. So in this picture, these are all 15-year-old girls. All of them are the same age. And this girl has pituitary dwarfism. And so the question is, what causes pituitary dwarfism and how could you possibly treat it? Okay, so the cause of this, dwarfism actually has many different causes. Probably don't realize that, but there's many different ways to sort of stunt growth. But one way with this is that you can have a mutation in the human growth hormone gene so that it's not produced. So in the case of this pituitary dwarfism, it's a recessive, autosomal recessive. So what does that mean if something is recessive and autosomal? Do any of you remember? Yeah, definitely.
Recessive, you need two alleles for it to be expressed. And autosomal means it's passed through. It is a part of the body. So
Yeah. So it's not on a sex chromosome. So on one of the other chromosomes. So for this, pituitary dwarfism is created from basically a mutation that causes very little protein to be made. So you would say that this, since there's very little functional protein, it's a null allele. I'll just write that down. It's a very severe mutation. This means there's no protein made. And so if you have one copy of this mutated version and you have the other copy of the unaffected, I don't like to use the word normal, so I always say unaffected allele, then you have enough. So even having half the amount is enough to get normal growth. But if you have two copies of just of this affected allele, you don't produce any, so you're going to have stunted growth, right? You'll have slower growth, shorter stature, and they realize this, they can start detecting it very early on. So I don't know, you all probably don't have children, but when you have children and you take them into their appointments, they always show you the growth charts. They show you these curves, and they're like, okay, you know, they tell you their height, their weight, and their head circumference, which is funny because one of my kids had the biggest head. It's just always had such a huge head. But anyway, they'll say like, oh, they're 50th percentile or whatever. And you kind of just track them as they go through their appointments. So they can catch this very early on. And if they notice like, all right, your child is starting to go, you know, it's not following the growth curve that is expected, then they might be concerned. So for this, it is entirely treatable if you catch it early enough. And you can treat it by taking growth hormone. So you have to supplement because you're not producing it. You have to take that hormone therapy before the closure of the growth plates, because after that, development is kind of already completed. So the approach here, if you wanted to get this growth hormone, how do you produce it? That's the first question. Where could you find human growth hormone as a source? Because you obviously, you have to give them fully formed protein as a therapy. Any ideas on where you could source that from?
The pituitary gland?
Yeah, so the pituitary gland of who, or
Oh,
of where would you,
Normal.
yeah, you're right, pituitary gland.
not normal, unaffected.
So maybe you could, so that might be difficult though, just, you know, it's taking a protein from the brain of a living person, right? So in the very early parts.
Would you think to the bloodstream though?
That is, yeah, I guess that's true, it would travel through the gross. But also there's sort of a regulation of when you produce the growth hormone, right? It's not produced throughout your lifetime, it's produced early on, so you'd have to take it from children. So that's an issue. They used to take it from cadavers. If they would source out some from cadavers, but obviously there's problems with taking proteins from dead individuals. And so there's another way to do this, which is that you don't have to produce it at all in a human. You can just take the gene from the human and you can make a bacteria do it, produce it for you. So that's the approach, is to clone the human gene into an E. coli vector and just put that into bacteria. Bacteria will pump out the protein for you, and then you lyse the bacteria, and all you have is a whole bunch of that protein. You can purify it so that you're only taking that protein and not other proteins. And then you could use that as a supplement, right, as a therapy. So if we want to do this process, we have to obviously put it into, we need to put that human gene into a vector. So I'm going to talk a little bit, you know, what is needed to do this. So in this case, we're going to put human growth hormone into a plasmid. Some of the tools that we need for this process, we have to be able to cut. So the molecular scissors are restriction enzymes. They cut at a specific sequence. What does ligase do? We've already talked a lot about ligase. Yeah, it's going to seal these pieces of DNA together. So we'll take, you know, one source of DNA, we're going to have to glue it together with the plasmid. And then we're going to put this plasmid into a host cell. A lot of times bacteria are used, but there's actually other, you can use insect cells to produce proteins. You can use, you can even actually put it into things like a goat. I'll give you an example of, they created this goat that expressed spider silk proteins in their mammary glands. And so they actually then could just milk the goat and they would get the spider silk proteins, which I guess is a very helpful material for doing like bioengineering for different like substances. It's a very sturdy material. So anyway, it doesn't have to be E. coli that you use as a host. So plasmids themselves, there's two different types that you can use. One is called cloning plasmid, and the other is an expression plasmid. The cloning plasmid purpose is just to hold the DNA. The goal is just, it's really like a holding cell for DNA. The goal is, in this case, is not to express. You're not trying to express the gene that is encoded in that. On the other hand, the expression plasmid, we can see that it has a promoter. The goal for an expression plasmid is actually to produce the protein. So in our case, which one would we want to use? Cloning plasmid or expression plasmid for this human growth hormone? expression, right? We need to put it into an expression plasmid so that the bacteria will transcribe and translate the gene. So the features of plasmids in general, we kind of talked a little bit about this with L440 plasmid. They're kind of the smallest of the vectors that we use. You can have bigger ones. They can hold up to 15,000 base pairs. So if you were trying to put something bigger, you would have to use a larger vector. They all have origins of replication. So what is the purpose for that? That was on our worksheet when we talked about L440. You can see the ORI here and the ORI here.
What was your question?
So what do origins do? What is the purpose for an origin of replication? Yeah, start for DNA replication, right? So that when the bacteria, because the bacteria has its own genome, and then the plasmid is over here, every time this replicates, we want to make sure it also replicates the plasmid. So the ORI is used for making sure the plasmid gets replicated. Most plasmids are going to have a selectable marker. This one has kanamycin resistance. This one says antibiotic resistance. It should say an R here to be a resistance. So what is the purpose of that piece? We also have that with L440.
So selection criteria, so that when we put it ampicillin and tetracycline into it, bacteria that we don't want to start growing.
Yeah, exactly. Yeah, so I mean, we have bacteria everywhere. We want to prevent the other bacteria from growing. We only want the ones that have our plasmid. They also will have multiple cloning sites, so we define that region on L440. It is shown here, so this little region right here is the MCS. You can see they have many different restriction sites there, so you can use different kind of molecular scissors to cut this open and put your insert in there. This does not show the multiple cloning site, but it would have been right here where the promoter was, and then there would have been something that you could cut on this side. So this has already been inserted in, so it doesn't show the multiple cloning site. Okay, and then I have that there, that the purposes of these are different. So cloning plasmids are just storing, just hanging on to DNA. These are often used when you're doing sequencing. If you're sequencing a genome of an organism, the idea is to cut the genome up, and you have to hold on to all the pieces. So PSmart HCCAN will be used, or others, to hold on to that DNA, and then you sequence within that plasmid. And then expression plasmids, like we said, they transcribe and translate the gene because they have a promoter. A lot of times they have lac operon features as well, so that they are inducible when you add IPTG. Okay, any questions so far on the plasmids? All right. So if you wanted to do this now, what are the steps that you would need to take? Well, first, you would cut your DNA sequence of interest. So in this case, we're talking about human growth hormone, right? So you would want to cut, and we'll talk in a second about how do you get the human growth hormone, because obviously, you know, that has to come out of humans, so you have HGH. You would want to cut this with a particular restriction site, with a restriction enzyme. Okay, and a lot of times when you clone, you use two different ones. And the reason for that is that way you can actually get this to go in only one direction. So you would cut, you can either use the same on both ends, or you could do two different ones. Then you have to cut your plasmid. Here's your plasmid that's sealed up. You would want to cut it with the same restriction enzyme. Restriction enzyme 1, restriction enzyme 2. So you can see what would happen here is this is going to just open up, and it would have sort of the compatible ends for the restriction sites that you used. And so they can then seal together with this. Then you combine those pieces together, and ligase will seal them up. So it seems simple. there's a lot of like tricks to it, and it, you have to figure out the right amounts, how much plasmid do you mix with the DNA, so there's a little bit more, it's more nuanced than what it appears, but those are the basic steps. So here's what that looks like in a picture. This, when they show this in your textbook, they show one single restriction enzyme. So here is the restriction site. That's the sequence that this restriction enzyme will cut. And the one they're showing, this is for, you don't have to know the name, but this is EcoR1. It's a very common one that is used. It searches for GAATTC, and it cuts right after the G. So it opens it up and it leaves sticky ends. So what might be the purpose of having those sticky ends? Anybody have any ideas? Why is this better than just cutting like you could cut right in the middle? Right between the A's and the T's, and just have a blunt cut. But what would be an advantage of having a flap? Oh yeah, go ahead, Daphne.
It leaves a DNA overhang, so then the gene that you want have can bind to the plasmid better.
Yeah, exactly. So you want to find a matching sequence, the compatible end, so it will only be able to bind to something else that has also been cut with that same restriction enzyme. So if you cut both the vector and the gene with that same restriction enzyme, then you'll create the same matching overhangs that this piece has to fit here. Now, I'm going to ask you to talk, maybe we should do this so that it's not as, you get a chance to talk to the neighbors, but with your neighbor, I want you to discuss what is different about using one restriction site versus two, and think about this with your human growth hormone gene that you want to clone. You want to clone it into an expression plasmid like this, so why would you want to use two restriction enzymes instead of just one if you were doing this cloning project? So talk to the person next to you to see if you can figure out why it's better to do two.
Oh no, wait. I feel like it's two because it would match up. I don't know.
There are more chance to success.
That's what I thought. I feel like if it's one, it's not gonna be. I don't know how to say it more. Like, if it's just one, it's gonna stick to one. But if it's two, it's more. I don't know how to say it more scientifically.
It's just more chances
Yeah,
to work.
yeah. Instead of one.
Double the success chance.
There you go. So yeah, there's always two sticky RE.
Yeah, I'm assuming for a given one you use a different restriction
Yeah.
and a nucleus. So when you just like, they both try to combine to the same sticky ends.
Okay, so how about somebody from this side, a representative, you want to share what you're discussing? Why is it better to have two if we're trying to, in this case, we're trying to clone human growth hormone into an expression? Yeah, go ahead, Amy.
So that we can prevent the plasmid vector from binding back.
Yeah, that's definitely one benefit. So what Amy said is it can prevent the plasmid vector from re-annealing to itself. So if you look at this, one solution is just for this to go back to that, right? That would be a product that we don't want because it doesn't have the gene of interest in there. Can anybody think of another reason? In addition, that's one very good reason. Are there any other reasons why two is better to have than one?
More cloning.
What did
More
you say?
clones.
More clones? Well, it's actually harder to get the thing that you want to anneal. But I want you to think for a second about transcription. And think about like from your exam where you looked for a gene sequence. And you looked at the top strand of the DNA, you looked at the bottom strand of the DNA. But for the promoter, so remember with our plasmid that we're trying to put into, you have a promoter going in this direction. Why would it matter? Why should I have two ends versus just having the same on both ends of the gene? Yeah, Daphne?
Is it so you're able to get another promoter on the other side to start coding for another strand?
Yeah, so I see, yeah, you're thinking definitely about the RNAi project where we had two promoters, so I can exactly see what you're thinking. In the case of expressing something and we only want it to make one mRNA, we only have one promoter. But as you can see here, if this is the flap and this is the flap, this is the 5' end of the gene, that's the 3' end of the gene. In theory, it can go that way, but it could also flip over, right, and go in backwards. And if it goes in backwards, you basically have the wrong strand that is next to the promoter. So the gene is not going to be read the same, right? You basically have the template strand flipped in. So in order to make sure this goes in only one direction, if you have one set of ends right here that only match up with this 5' end and a different flap on the other side, you can make sure that it can only go in this 5' to 3' direction. Does that make sense for everybody? So there's kind of two benefits. One is what Amy said, which is that it prevents the vector from just re-annealing, which is not helpful for us. And the second is that it makes sure you get directional cloning. So I'll write that word, directional cloning. You only want the gene to go in one direction. Okay, so a little bit more about restriction enzymes, since we looked at that EcoR1. Question is, where did these things come from? Did we create them or what? They actually were found naturally occurring. So bacteria have them, and people just discovered, wow, bacteria make enzymes that search for a certain sequence. Now that seems a little weird. Why would a bacteria want to chew up DNA? And isn't that a risk of it chewing up its own DNA? They were developed, or it's thought that they were developed evolutionarily to be a defense against exogenous DNA. So like viruses that come into bacteria, the viruses will get chewed up by these naturally occurring restriction enzymes. The reason why bacteria don't chew up their own is typically because, remember how we talked about bacteria methylate? Remember how we said when we were talking about mismatch, that one of the ways that they protect their own DNA is they methylate it. So that a lot of these are methylation specific, that they won't cleave methylated DNA. So they're only going to cleave DNA that comes in from outside. With these different ones, they typically recognize a palindrome. So palindrome means it's the same forward as it is backwards. And they typically cut in a characteristic position. So they can sometimes create sticky ends, either a 5' overhang, or it can cut and produce a 3' overhang. If it cuts in the middle, right through here, it will create blunt ends, and those also do exist. Those can be useful if you're just trying to like get stuff to go together and you don't need it to be a specific pair up. So their blunt end enzymes are sometimes used for certain purposes. But the really important thing is that for us, we can use two different ones to get directional cloning of a gene of interest. Okay. So let's talk now, back to our problem here. We're trying to get human growth hormone. We want to put it into bacteria. So let's talk about where do we get that gene from? We said that we know that it is produced in the pituitary gland. But the question is, if we're gonna go get source DNA from humans, which type of DNA should we get? So bacteria, let's just for a second think about what are the differences between bacteria and humans. Are there any differences in the way that they transcribe and translate?
Humans can have a lot more space to do those where bacteria are like really condensed.
Yes, that's one difference. So their genome is more compacted than ours. So what is one of the things that our genes tend to have inside them that make them a lot longer? Introns. Okay, so that's the big thing that we gotta think about for putting human growth hormone into bacteria. We can't put introns into bacteria. They don't know what to do with them, right? So we have to somehow get a version of the gene that does not have introns. Now that seems like that's gonna be kind of a tough thing. So we cannot use genomic DNA. If we were to go and take genomic DNA from humans, we could isolate that pretty easily, but it would have the introns, and that's not gonna be tolerated by bacteria. So we're gonna use something that is called cDNA. You probably have never heard of that, and that's okay. cDNA stands for complementary DNA, and it is DNA that we actually produce from the mRNA. So we start with the mRNA, and we start with mature mRNA. So if you think about, in humans, we have DNA produces the pre-mRNA through the process of transcription, and then there is splicing that produces the mRNA, right? So by the time you get to mRNA, there's no introns in this, and it reflects exactly how that cell would have done the splicing, whatever kind of alternative splicing patterns would have been. They've already been spliced out, so you can see exactly how it was spliced. The way that you make complementary DNA is you actually go from RNA, you reverse transcribe it. So you go back to DNA from the RNA. So this is something that we as humans have to do when we want to make cDNA. It's not naturally occurring. We reverse transcribe it back to DNA. We can make a single strand, and then we have to make the second strand. So essentially what we do is we create the way that this was spliced, and now it's back to DNA form. So let me show you a picture to kind of show you what this looks like. First, let's look at genomic DNA library. If you're going to make a collection of all the genomic pieces from an organism, you would randomly cut it up. So when you're making a genomic library, you use restriction enzymes to cut. You can see that when you do that, you sometimes cut in the middle of a gene. And then you take these pieces and you put them in vectors. So this, obviously, you can see this contains all of the non-transcribed DNA. It has introns, and it cuts up genes in the middle. So that's not going to be useful if we want to isolate the human growth hormone gene. When we get cDNA, we actually start with mRNA. So you go to a specific cell type, you collect all of the messenger RNA from that cell, and then you reverse transcribe it, and you can see that the only thing that's going to be here is exons, right? So this shows you exactly how this was spliced, and now you have only the exon regions, and you only have the genes that were turned on in that cell. So for this, gene B was being expressed at a high level. You have a lot of copies of it. Gene A was expressed at a low level, so there's only one copy of it. Does that make sense to everybody? All right, so here's your concept check. You can talk with the person next to you. Which one of these would we want to use for our insert? If we're trying to make human growth hormone, where should we go get it?
cDNA particularly that,
Yeah.
because it has all
cDNA
the
has,
expressions of the protein that we're trying to get.
yeah. Because the genome has introns and so it does not.
Yeah.
Okay, so which one are you leaning towards? What do people think? See? Everybody agree? Yeah, so we have to have cDNA because we can't have introns. And then we also have to go to a cell that we know makes that protein. So pituitary gland is the source, so that would be where we would want to get the cDNA from. Okay, I have a couple of slides that show you when we say genomic library, why is it called a library? And cDNA library, why are they called libraries? So essentially a library is a collection of all the pieces of DNA, either from a whole genome or from all of the messenger RNAs that were being produced, if we're talking about a cDNA library. So we used to, in this class, this was for years and years, I think it was like 10 years, they did this project where they were mapping the genome sequence of this organism, which is a type of bacteria that's, I guess, very useful in eastern Washington for protecting against a certain type of fungus that overtakes these wheat fields. Anyway, so that's what the students used to do, is they actually would take a clone that had a piece of the genomic sequence, and they themselves would do the genome sequencing reaction. So to make a genomic library, you basically take all of the DNA from an organism, either bacteria, or in this case they're showing human, you cut it up, and then you put those pieces into individual plasmids. And then each plasmid gets inserted into a different bacteria. So that students in that class would pick a clone, and they would sequence their portion of the genome, which would be one tiny little stretch. So this is a genomic library. The library lives as a collection of those bacteria, and every single one has a different piece. Now that's different than cDNA library. So obviously we have to construct the DNA from the RNA. So first question is, if you're going to start with mRNAs, how do you make sure that you're only getting messenger RNAs and not like tRNAs or rRNAs? Any ideas what you could use to isolate just messenger RNAs? So for this, I'll give you a clue to think about. What does messenger RNA have that those other RNAs don't have?
That's cap and poly.
Yeah. So you actually use the fact that it has those end pieces. Then one of the ways you can do this is you can use we know that it has a poly A tail, right? So all of your mRNAs have this long string of A's. You can use a bead that has T's on it. So you can use an oligo DT column. That's basically a bunch of beads with oligo DTs. And essentially when you run all of the RNA from the whole cell, you basically run it through a tube. Just if you guys remember the plasmid purification, how we had that little column, you could put these beads in the column. You could run all of the RNA for the whole cell through this. The only thing that would stick would be messenger RNAs. So to answer that question, how, you could say that you can isolate the mRNAs using their poly A tail. And then, once you have those collected, then you can synthesize the cDNA using reverse transcriptase. So reverse transcriptase we get from viruses. We just isolated it out of viruses, and we can actually use it in the lab. It's going to build a single strand piece of DNA. And then we can chew up the RNA and synthesize the second strand. So we know that DNA is most stable when we have two strands. So once you have that piece, you put it into the vector, and you transform those plasmids into the bacteria. So that's what a cDNA library would look like. Okay. I guess I think we can save this for tomorrow. We'll start with PCR. And that will actually start with, we'll use this, we can do this activity tomorrow. So why don't we take a little 10-minute break? I just feel like I'm roasting and everybody's so tired. So we'll come back at 12:45 and then we'll start working on our chunk